Okay, so the next talk is from Professor Dr. Evangelos Manalopoulos, the Chairman of the Board of Directors of EOF. He is the Professor of Pharmacology, Pharmacogenomics and Precision Medicine, and the Director at the Department of Pharmacology of the Demokritos University of Thrace Medical School in Alexandroupolis, Greece, and at the Clinical Pharmacology Unit of the Academic General Hospital of Alexandroupolis. He obtained a PhD from the Medical School of Patras University, and he did postdoc at the University of Wisconsin. His current interests include research and clinical applications of pharmacogenomics and epigenetics in drugs used for cardiovascular diseases, diabetes, anticoagulant therapy, and psychiatric diseases. Dr. Manalopoulos, the floor is yours.
Okay, good morning. Good morning, everybody. First of all, I would like to thank Professor Patrikios for inviting me and to congratulate him also for organizing one more excellent European University of Cyprus Congress. It's always fun and pleasure and scientifically interesting to be part of this endeavor in this meeting. Now, today I will talk to you as a professor in university, not as a president of the National Drug Organization, and I will present you our work on anticoagulant therapy, which spans several years, so just some snippets of it. And you can see here in the hospital in Alexandroupolis. And, well, this is an old story starting from Hippocrates already 400 before Christ, year before Christ. He coined the term thrombus, and already by that time there was suspicion that by thinning the blood, and later on other philosophers and scientists were about the same at the older times, was suggesting that thinning the blood may improve certain conditions or prevent certain conditions like thrombus formation and what we know now as thrombotic event. Well, I'm go through that. I will not analyze that. This is from how he perceived the body and the fluids of the body and how correct many of the things he perceived were, well, keeping the analogy. Now, coming to the modern era of anticoagulant drugs, starting from the 50s already, when warfarin and the other vitamin K antagonists were at first discovered and used and saved millions of lives worldwide. Now, the field, and there were some problems with them. They're difficult to stabilize. They have a large heterogeneity in their effect. They interact with other drugs, with food. So they were very effective drugs, difficult to use. So there was a need for newer drugs already at the end of the 21st century, the first decade of the 21st century. They started emerging, first dabigatran, and quickly after rivaroxaban, apixaban, and doxaban. This is kind of the situation. We have 4 new direct oral anticoagulants. This is the situation. As we said, warfarin is a vitamin K antagonist. The other drugs are directly inhibiting either factor 10 or thrombin, as you see in the slide. And as I said, there is heterogeneity in the effect of vitamin K antagonists. A lot of research is taking place there. One of the major areas where pharmacogenetics has been developed was on uncovering genetic factors contributing to this heterogeneity, and it's been established now that cytochrome CYP2C9 and vitamin K, an enzyme involved in vitamin K metabolism, are heterogeneous, and they have phenotypic diversity, and they contribute to phenotypic diversity. Several other factors have been developed. Only the genetic factors contribute about 50% to this variability. So we have made strides there in characterizing the pharmacogenetic variability. We have been fortunate to participate in a European consortium that released the first randomized clinical trials on this issue back 10 years back in New England Journal of Medicine, where sort of contributed to establishing indeed that these polymorphisms are involved. But, well, and then the field developed further. There were more things coming out that you need ethnic-specific algorithms, for example, and we have several years ago published a Greek algorithm as it emerged that you need a specific, ethnic-specific algorithm to better modify the drug therapy. So we published that, but, well, sort of all this became irrelevant, or more or less, because the new drugs have taken over the practice of medicine. Whether that's fair or not, it's another discussion, but somehow very few specialists now are using vitamin K agonists. Mostly it has been moved to direct the DOACs. So we and many others shifted our attention in looking at the DOACs and finding the heterogeneity there, and there's some studies on the different on the pharmacogenetics of DOACs. I will not go into them, but then omics are coming into play more and more. The personalized medicine that you've been hearing a lot about today involves looking at all different levels, not only at genomics, at genetic level, but also transcriptomics, epigenomics, proteomics, and other levels, even more specific, like metabolomics or phosphoproteomics. And then the field is emerging from going from a single biomarker, like the cytochrome I was telling you, to a signature set of biomarkers that are predicting the right response and adverse drug reactions, and then even networks that are involved in these processes. So we've been interested in that, this multi-omics approach, and we created a project, several projects to look at the epigenetic contribution to the diversity of responses to anticoagulants and to atrial fibrillation. Just here to remind students here that when we talk about epigenetic, mostly it's about DNA methylation, histone modifications, and microRNAs, which are, well, practically they're not modifications of DNA, but they also contribute indirectly to DNA expression regulation. And in principle, when a gene is methylated or hypermethylated, it's turned off. So it's another way to control. And microRNAs also have regulatory functions on the expression of genes. So looking what has happened in the field of coagulation and DOAC treatment at the epigenetic level, very few things have happened. However, there are some indications, for example, that at atrial fibrillation there's epigenetic factors contribute to atrial fibrillation. Not much, or if anything, has been done on the different DOACs and how they affect their control at the epigenetic level. So we established a clinical protocol entitled Circulating MicroRNAs and DNA Methylation as Regulators of DOAC in DOAC response in atrial fibrillation. And actually, our goal and the goal of all the projects we have in this field are twofold. One is to identify biomarkers of the DOAC response in total, but also biomarkers of heterogeneity between the three different drugs used, because we still think there might be differences, although they're treated by clinicians now as one uniform group of drugs, and then they look at different clinical factors, how to choose which of the three different ones to use. We think that maybe there is some under background, some genetic or epigenetic background that could help specify the dose and target the dose better. So this is the idea behind our project. So actually, the design of the study has been published already, and two departments are contributing the samples from Athens, Professor Trika's group, and in Alexandroupolis, the cardiology clinic from Professor Giakas. And that's where we are so far. The idea is we have newly diagnosed patients with atrial fibrillation, so we sample them before starting therapy, a week after starting therapy, and 4 weeks after starting therapy for all three different drugs, and we have also a control group. And we've looked at methylation, but I will not talk about the methylation results today. I will more concentrate on the microRNA results and some proteomic results if I have time. So our approach is we're using the TACMAN assay, which includes 754 microRNAs, and we studied them at the same time on this scientific platform. So we can see, essentially, these are most of the RNAs known to be involved in human reactions. So we look at the myrinome of the organism. Those are the 12 patients at which we have looked at initially. This is an agnostic approach to see what microRNAs may be affected by the drugs. Of course, they have other diseases, and so we are analyzing also other phenotypes, but looking at the comparison here, we find that each drug drives different changes in myronome, and so we have rivaroxaban, dabigatran, apixaban. You see anything that's top left above the line is overexpressed. Anything that would be above the line but on the left is underexpressed, or on the right is overexpressed. And we're analyzing these results. Well, when we pull together for all the three drugs and analyze, we find 2 microRNAs, and we correct also with benzamini fast discovery rate. We find 2 microRNAs that are significantly correlated with DOAC response, the microRNA 204-5P and the 20-5P. Well, then when we use the remaining patients as a validation cohort to see if this difference holds, and we find for one of them, actually the microRNA 20A, that this indeed holds, so there's a 1.4-fold increase in all the cohorts, so we think there we have one potential marker for a DOAC response, and microRNA that is involved in the effect of DOACs. And looking at what this microRNA does already from the literature, we see that it's involved with regulates several of the coagulation pathway factors like apolipoprotein 3, the receptor 3 and 4 for thrombin. So it's an interesting molecule to analyze further. And for the other microRNA, we did not, at the validation cohort, there was no significant increase, but we noticed something else interesting that we're analyzing now, that there was difference between the different DOACs used. So in the case of rivaroxaban or apixaban, the direct factor 10 inhibitors, we see an increase, which is not statistically significant. But in the case of dabigatran, we see a decrease. So when you compare factor 10 inhibitors with thrombin inhibitor, there's a difference, and actually in different directions. So this is a hint of heterogeneity in their effect, which we analyze further, because, as I told you, on the back of our mind, heterogeneity in responses is something we're interested in. Well, a few things about our proteomics approach at the same cohort. And you see proteomics is going for, you can fish many fishes at once and not just, well, the guy on the left has a better time, I'm sure, but the guy on the right is much more productive, and this is the case with proteomics approach. And it's important because if DNA tells us what could happen, RNA tells us what very possibly will happen. At the protein level, when we find something, we know what actually happens as a result of the previous steps. So the project to look at the proteomic level is to look at endothelial cell cultures, HUVECs, and then we stimulate them with DOACs and see how the different expression protein at the protein level. Of course, we do the same also in we do proteomic analysis also in other projects, but this is the one for the different DOACs. And well, this is the protocol. I will go through it quickly as time is finished. This is all the protocol we do, the proteomic approach. And then we look at common proteins expressed by 4 DOACs. And we have, if you look at this, 4 proteins that are commonly overexpressed after DOAC stimulation. And when we start looking at them, what they do, well, at least one of them, this APP, is an important regulator. Well, the RSU1 is correlated with APP, which is an important regulator of vein thrombosis and controls coagulation. And it is, interestingly, it's also regulated by MIR20A, which I was talking before. So we are also looking this, how these pathways connect at the 2-dimensional level, because it is a multi-omic correlation. And then we have these 4 proteins. When we look at the literature, they have anti-inflammatory, cardioprotective actions, anti-stress, anti-fibrotic, anti-atherosclerotic. But so we are looking at potential pleiotropic effects of DOACs here, and what we find so far is mostly on the positive level, so it seems that drugs have overall, we're enhancing a positive effect on cells, which is also the clinical experience so far. And then underexpressed proteins, we have found 14 that are underexpressed in all cells. Again, with the bioinformatics analysis, we pinpoint out potential proteins of interest to study further. So, well, to summarize, this is a multi-omics integration for precision diagnostics that we are looking at, using all these omics connected together, co-analyzed with the clinical phenotypes, environmental factors, all the profile of the patient, and then this could lead to endotype-based diagnosis, different subgroups based on all the factors, and also omics. We don't say omics is the most important. We think omics can contribute at several levels together with other clinical factors that are already known or are being discovered at different fields. So the idea is that all this information goes in the system, and then correct diagnosis and correct prescription comes out, and this is personalization at the clinical level. We have expressed our ideas recently in a commentary with my colleague, George Raya, and I think this is, I'll close here. I say that much of this work is part of the ongoing PhD work of Mirya Palikar, who is in the audience also, and has an interesting poster with our work on the proteomics at the atrial fibrillation level at a different project. So if you're interested in proteomics, please be invited to talk with her. And I would like to thank all my collaborators. Thank you.
Thank you, Dr. Manolopoulos. Questions from the audience? No? We have one question.
Thank you, Vangelis. Would you please kindly comment on the clinical applications, daily clinical applications of the various pharmacogenomic testing, because there is a hype on the use of these assays, and what would you expect in the coming years?
Well, yes, this indeed, if we look at what is happening with pharmacogenomics, which is a simpler concept, it's still, and there's established biomarkers, for example, clopidogrel, CYP2C19, still there's resistance at the clinical level to be accepted. So if you look at that, one should be very pessimistic on what will happen with these omics markers. However, I think at some point there will be a shift in the mentality at some level, and all these accumulated markers, genetic markers, will somehow get incorporated into the clinical thinking. And I know, for example, countries like in Spain, I was recently there, that they have a nationwide initiative to implement pharmacogenetics for all patients through the hospital system for several years, and to confirm at the real-world level that this is effective, reduces ADRs, is cost-effective, and such approaches may generate data at the level of the ministry, not just what we're talking here, but this is at the level of the Ministry of Health in Spain, at the National Drug Organization also, and that's why I was there. So I think there has to be some approach from top now. There's a lot coming from the bottom. We need something now to come from the top, either from the state or health insurance companies, national or private, or from medical societies. But there is resistance at the medical society level, cardiology. In oncology, it's even worse for several markers, for example, tamoxifen and CYP2D6, that 15 years now, they are resisting to include it in their guidelines. So, well, I'm optimistic by nature, so I expect something will happen sooner or later on this. Thank you, Achilles.
Okay, thank you very much. So, the next talk will be rescheduled for later by Professor Ramia Keilan. There will be no coffee break, and we will proceed with the next session.
We would now like to invite Dr. Georgios Ioannis and Dr. Maria Eptaka and Dr. Christiana Gusparou to chair the continuation of this session.
So good afternoon, ladies and gentlemen. My co-chairs and I welcome you to this next session titled New Frontiers, which covers a wide range of topics on cancer treatment, including immunotherapy and integrative approaches. Unfortunately, our first guest speaker couldn't make it, so we're having only two presentations today. So, Dr. Eptaka, would you like to introduce?
Good afternoon, and from me. I would like to introduce Dr. Konstantinas Stylianou. Dr. Konstantinas Stylianou is a pioneering integrative oncology specialist who has made significant strides in the field by establishing the first integrative medicine department in Cyprus at the German Oncology Center. As the official ambassador for integrative oncology in both Cyprus and Greece, Dr. Stylianou is dedicated to advancing integrative cancer care approaches. Her academic journey began in London, where she completed her studies in Chinese medicine. She furthered her experience with a master's degree in nutrition and earned her doctoral degree in the United States, specializing in integrative oncology. Her lecture title is The Science of Living Well with Cancer, the Role of Integrative Oncology in Cancer Care. Dr. Stylianou.
Is Dr. Stylianou here? No.
Okay, we will try to find her, possibly for her to give us her talk later on in this session, however. So if you guys agree, it gives me great pleasure to invite to the podium Professor Agamemnon Epenetos, who is my scientific father, which is different to biological father. Professor Epenetos has been my tutor for almost 30 years now. We have together worked in cancer and stem cell biology, both at Hammersmith Hospital, Imperial College, and also now in Cyprus. I would like Dr. Ioannidis to share a little bit on his CV, and then we can have a discussion at the end. Thank you.
Thank you. So let me remind you that Professor Agamemnon Epenetos qualified in medicine from Glasgow University, subsequently specialized in medical oncology at St. Bartholomew's Hospital in London. He was offered a fellowship at the Royal Postgraduate Medical School, Hammersmith Hospital, Imperial College London, as a consultant and professor in medical oncology and cancer research. He has been a practicing oncologist and cancer researcher over the last 30 years and a visiting professor at Imperial College London. In 1992, and together with Dr. Achilleoudi, he encouraged senior members of the government of Cyprus and the Bank of Cyprus to create and fund the Bank of Cyprus Oncology Center in Nicosia, which is a state-of-the-art treatment of cancer. In 2022, he was offered and accepted the position of Director of Research and Development of the International Stem Cell and Regenerative Medicine Research and Therapeutic Center in Cairo, briefly called Egypt Center for Research and Regenerative Medicine. So Dr. Agamemnon-Ependitos has more than 20 years of experience as a biopharmaceutical industry executive. He founded and served as chairman and R&D director of Antisoma PLC, Lipoxene PLC, can move on. Yes. So Dr. Ependitos has received much international academic recognition and awards, and his very anticipated lecture is under the question entitled Immunotherapy of Cancer, a Dream Becoming Reality.
Well, thank you very much. I don't like this history of many years. People may think I'm old. First of all, I'm very grateful to Professor Ioannis Patrikios for giving me the opportunity to present to this wonderful conference. And I didn't know what to expect. I was actually slightly confused by the word multi-thematic. It sounded sort of a mixture of Latin and Greek. So I Googled it. I didn't actually Google it. I took one of these new apps where you type in a title and it gives you a picture. And I got this picture. Oops. I got this picture on, I need to, go back a slide. And I'm told I'm pointing at this. That one, okay. Yeah, I got, oh yeah, you can see that. And this is the printout from the internet. Obviously, they're way ahead of intelligence from us because they are telling me that instead of being multi-thematic, it's actually intergalactic presentation. So maybe Professor Patrikios one time, he can give a global can connect us so we can present all of this to China, not only some Chinese present it to us, and we'll make sure that the slides change. So let's start from the beginning. Oh, this is not quite the beginning. The beginning was a boy and a girl. The girl ate an apple, and she was asked with the boy to go to Earth, a planet which could have been paradise. But on Earth, as you know, we have a lot of evils. And for the benefit of this conference, I'm focusing on cancer. I keep pointing. I'm sorry. I'm used to using a laser printer, which is, this is changing, yeah. Anyway, I will talk about it. So we're on Earth, they found all these terrible evils, but Pandora eventually managed to close the box and a little devil was locked in. And that devil was called Elpis, Elpida in Greek, hope. So my starting point is that never lose hope, especially if you're involved in research and development. Hope, I equate to a dream and expectation. And really what I'd like to present to you for a few minutes is this, wouldn't it be great if we could proactively educate our immune system to attack and kill cancer? We already know that our immune system is doing that job against many infectious diseases, and although we use antibiotics in parallel, but you need to use a healthy immune system as well. So really, this is the dream I'm presenting. But also, it's not my dream. It's been around for thousands of years. Last night, we heard about the Egyptians and the genetic family of Cleopatra. But 3,000 years ago, the Egyptians made very interesting observations that tumors could disappear if the patient had an infection with a high fever at the same time. Perhaps it was the first example of hyperthermia in cancer. And many things happened at that time in Egypt. But I think the most important thing is discovering papyrus that we could turn it into paper so they could document all their data rather than keep writing on the walls these hieroglyphics, which now, even nowadays, some of them haven't been interpreted yet. So it's good to document your data. And more or less at a similar time, a very impressive Greek physician, Galen, who was not only a physician, but he was a philosopher, he made this very interesting observation and also tried to connect that cancer may evolve from inflammatory lesions. I will show you one slide next to perhaps mention that. But the father of immunotherapy was neither an Egyptian or a Greek, unfortunately. It was an American. And worse than that, he was a surgeon. And this American made the same observation, William Bradley Coley. He's the father of immunotherapy. And he was the one who actually made the same observations, but proactively he took bacteria from an infection and started injecting them into tumors. Some of them were also inactivated bacteria. But he observed some changes, some improvements. So I would argue that this is the starting point of cell therapy against cancer, although they were bacteria, they are still cells. And there's a negative control. You can use the bottom line. About 1 in 100,000 people, the cancer disappears spontaneously, which is, again, is a slight mystery. But by the time we discovered those patients, the cancer has disappeared, so we cannot do anything about it. So to go back to Galen, he might have been a fantastic molecular biologist if he was nowadays, because this is the observation that we see when we use this family of transcription factors called NF-kappa B. You can see it's connected to inflammation, and it's also connected to important cancer steps. So there is some connection, and obviously we need to disconnect it, because inflammation, we can eliminate it. The cancer, some of these changes are actually permanently switched on. So to jump into nearer this time rather than a few thousand years ago, for the last really few decades, there have been dramatic progress, in understanding the genesis of cancer, immunobiology, and allow us to have a more scientific immunotherapeutic approach. So believe it or not, we're now living at the beginning of a new golden era in biomedicine. Despite all the world having all these troubles around us, this is a good time to be a doctor and a researcher. So cancer immunotherapy is fascinating because it's really the product of reciprocal learning. We're learning from the immune system how it works, and the immune system is telling us, and we're telling the immune system what to do. For the purpose of this meeting, I've divided my talk into 4 sessions, which should take very few minutes each. One is cancer vaccines, as you can see there. Cancer vaccines, I'm using the term as a vaccine. It's a vaccine which you give to somebody to prevent them from developing a disease. There is small parenthesis in there. You can have therapeutic vaccines. You can give something that is a vaccine, but the patient already has a disease. Secondly, I'm going to talk about monoclonal antibodies, which is an area of great interest to us. We're the first people to show in the world that these things work. And checkpoint inhibitors, of course, is a family of antibodies. And finish with CARs. I know there is the Grand Prix around, but it's got nothing to do with CARs. So, vaccines. We have 2 wonderful cancer vaccines. One is hepatitis B vaccine. It was the first anti-cancer vaccine. And this can treat probably one of the commonest cancers in the world. Liver cancer is very common in China and in the Far East. It can be eradicated, prevented in more than 80% of patients that will develop the disease. Hepatitis B is a very important anti-cancer vaccine. Followed by the HPV vaccine, human papillomavirus vaccine, which it was licensed more recently in almost all over the world in 100 countries. And this is recommended for young children, young adults. Then it should be recommended for both boys and girls, although the main disease that you get is cancer of the cervix. In the boys, it prevents the very rare disease of penile cancer. So, and of course, again, it's been a fantastically successful vaccine, and 90% of cancer of the cervix has been reduced. This is the good news. The bad news, we have other important viruses that we haven't achieved a great deal. Very important virus is the Epstein-B virus, which is a latent infection. It was originally discovered when people were studying Burkitt's lymphoma in Africa, but is now linked to a multiplicity of different lymphoproliferative lesions, malignant lymphomas, etc. There is also evidence that is triggering multiple sclerosis. Sorry, I'm moving towards the screen, but I'm moving away from the microphone. which is very microscopic. I can look something down. And if, although with great pomp and circumstance, a company called Moderna was spun out of Harvard a few years ago with the main purpose, they had the technology of mRNA vaccines, and they wanted to focus on EBV virus. Hundreds of millions of dollars have been spent, but the last time I looked in May, in publications in May 2024, they are still at preclinical stage. I don't feel sorry for Moderna because they made billions from selling the COVID vaccine anyway. But it would be important to have an EBV virus. But it's a fascinating virus because, as you can see on the bullet point of this, it's a dual tropic virus. It infects both B cells and epithelial cells. So, interesting virus, but we're not succeeding on that yet. I will jump in now into an area that I know very little, but is very, very exciting. I thought I knew everything, but I know very little in this area. And when I was doing research earlier years and looking at various vaccines, we were looking initially at the lower paragraph, we were looking at tumor-associated antigens, because you could see at the beginning, there are some antigens, like carcinoembryonic antigen, polymorphic epithelial mucin. There are various cell surface antigens that are expressed at low levels in normal cells, but they are highly expressed in cancer. And we thought if we can try and make vaccines against these tumor-associated antigens, we would be successful. But we're not really very successful because, first of all, an immune response doesn't really depend on the amount, to some extent, of the antigen. In other words, you cannot discriminate between a low level of antigen in a normal cell versus a high level in the cancer. And that you run the risk of either not switching on an immune response, or if you switch to one, you could also have an autoimmune effect. However, the neoantigens are a different story altogether. They are foreign proteins, totally foreign proteins, developed by a patient, which the immune system does not address those antigens. They just come on when a tumor, for example, has mutations or goes through various omics, as we said here, but they are neoantigens. They are proteins that are unique to that individual patient. So my definition of individual therapies is something like a neoantigen. These are unique to each patient. So how can you do something for each patient? The next slide is another small parenthesis, because I work mostly in the UK, and I worked mostly in National Health Service Hospital. But what was fascinating, the NHS, which the equivalent in Cyprus is called YESI, the NHS, which controls the budget of everything medical in the UK, they had a thought. I never thought that these big organizations have any intelligence. But they had a thought. They wanted to go again into the story, prevention is better than cure, and also for the NHS, prevention is cheaper than cure. They were really money-driven. So they formed this amazing NHS Cancer Vaccine Launchpad, CVLP, and the main purpose of that for the NHS, think of YESI. Can you see the YESI go there and say, we're now going to develop vaccines against neoantigens. So they took the idea that we're going to use mRNA to develop individual vaccines for every patient. And they thought this will accelerate cancer vaccines as a form of cancer treatment. And they've already taken aim because they they hold the budget. If somebody holds the budget, you do what they tell you to do. They control 30 hospitals in the UK. They're already pioneering cancer vaccines. And of course, what I didn't tell you, they are doing all of this in partnership with industry. The two companies they are working with is BioNTech from Germany, who have the experience of mRNA vaccines, and Genentech from the USA. So there is a partnership of private industry together with the National Health Service, and they're developing internally vaccines, individual vaccines for every patient. And of course, they think they will save a lot of money by doing this in partnership, but I actually think it's quite an achievement for the NHS to start thinking rather than just spending money, and I wish, yes, it could develop also this ability. In parallel, the US, of course, don't want to be left behind. The USA, they have Merck and Moderna doing much the same thing. Everybody's really jumping on the neoantigens. It's an exciting new topic, but let's think about it, because every time you have something exciting, there may be some problems in the process, but it's interesting. So the first topic, the second topic after the vaccines is therapeutic monoclonal antibodies. And again, we were some of the first to demonstrate that antibodies work in humans in vivo and in vitro, and therapeutically. Because in those days, we used to have polyclonal antibodies, and everybody kept saying, oh, if you get monoclonal antibodies, they will fall apart. The antibodies need to work as polyclonal. But also monoclonals work very well on their own. And the purpose of an antibody is to target to a cancer cell so that it can kill the cancer cell by drawing in the immune system or by behaving almost like a tyrosine kinase inhibitor, because a lot of the receptors are tyrosine molecules, and the antibody can block it, like the HER2. We still don't know exactly how much is immunotherapy and how much is behaving like an inhibitor. Also, they can carry drugs now. If you can now have like Herceptin, when it becomes a bit weak, then you can conjugate it into a toxic chemotherapy drug that is taken to the tumor, or you can conjugate it onto radioactive isotopes. So the antibody can be a carrier of a therapeutic drug. And they are very effective. You can also have bispecific antibodies that bind to the tumor and bind to the immune system, and that works. So in cancer, there have been more than 24, there are 24 monoclonal antibodies now on the market against 16 antigens, and maybe out of date because things happening quite frequently. Some of these antibodies, as you can see there, they are selling for more than $5 billion a year. So it's a big market, but obviously is the #1 leading therapy for cancer nowadays, monoclonal antibodies. The third part of my talk is the checkpoint inhibitors. If you look, first of all, at the beginning, this tumor cell here, you can see there is an antigen on the tumor cell, and we're now using T cells. We're now moving towards this theme in the next presentation in a minute. We use the T cells that can recognize, they could recognize even 20 years ago, the antigens that are present on cancers, and yet they couldn't kill them. There was sort of a blockade. And we used to recognize these molecules marked in blue that they had a role to play. We used to call them accessory or collateral molecules. We didn't know what they were, but a couple of groups, and I will show you in the next slide, were able to define that on the cancer cell, there was, in this interaction, there are so-called death domains. In other words, the T cell, to be able to do its job properly, must not bind to this blue receptor, because the blue ligand here developed by the cancer is like barbed wire. You cannot get through it, and that's the problem. So what has been done, which has been dramatically successful, is to develop 2 different antibodies, one that can bind to the PD-L1 ligand to break the interaction, or one that can bind to the programmed death receptor on the normal T cell. If you disrupt this relationship between these two, then you only have the T cell on the T cell receptor that binds onto the tumor, and that can kill the cancer cell. And this has been the dramatic thing in the last few years, actually, very recently. And 2 wonderful people, James Allison, who has recently moved to Texas, and Tasuku Honjo, they got the Nobel Prize for these observations of these molecules that now have this ability to kill the cancer. And basically, they inhibit the inhibitors. Therefore, the T cell can open up and kill, which tells us immediately that our normal T cells, without us doing anything about them, they have the ability to kill cancer cells. It's just that they're paralyzed by cancer. Cancer is a clever beast. Never underestimate cancer. Cancer is a clever beast. It develops all these inhibitors to inhibit the activators on a T cell. And that has been a great success. And also, what is very important, some of the patients that have been treated have been living very long. In other words, it's almost creating a vaccine effect around these T cells. The T cells, as they destroy the cancer, they may start having a sort of a longer-term vaccine effect. This is a fascinating area for further observations. And these checkpoint inhibitors, they're called checkpoint inhibitors, have been widely used in many different cancer types, but all these cancer types need to have the ligand on the cancer cell that can be blocked by the T cell. So you need to screen them to see if they're suitable for immunotherapy. This has been up to now. But again, a small parenthesis. If we now recently, and this is the work that was presented recently at ASCO in Chicago, up to now, we've been using one checkpoint inhibitor for one type of cancer. But what is interesting, at least from this particular study, which was published in Nature as well, if you take two checkpoint inhibitors, I'm not going to name them, I'll just call them Bot and Ball, two checkpoint inhibitors, they can now inhibit both the microsatellite stable and the microsatellite unstable colorectal cancer, because the ones that were unstable were originally responding to immunotherapy, which is a rare form of colon cancer, but the more common one were not responding. By combining two checkpoint inhibitors, it seemed to be getting very remarkable results, certainly measuring the tumor DNA in the blood, and the patients were, of course, at an early stage of treatment, appears to be very effective. So combining more than one checkpoint inhibitors could be synergistic. And if you look at checkpoint inhibitors, I've only showed you the reaction at the PTL level, which is up here. Joe Allison, who's gone to Texas, is now studying VISTA because he thinks VISTA is very important in pancreatic cancer. But look at all this list of checkpoints and checkpoint inhibitors. We haven't touched, this is the tip of the iceberg. We can make a lot more of these checkpoint inhibitors by dissecting all the parts of the tumor. So there is a great deal of hope, actually, I think, in this area with all these receptors. And the next slide, which is really towards the end of my talk, is the CAR-Ts. This is taking a normal T cell from the patient himself or herself, taking all the blood out, not all the blood, taking blood from the patient that contains T cells, genetically modify them in somebody's laboratory. Originally, it was done in Novartis laboratories. That's why they were selling them at the beginning for a couple of $1 million each. But you modify the T cell ex vivo, you expand them, and you give them back to the patient. So now the patient has its own T cells. It's almost reminiscent of coli at the beginning, as I said to you. It has its own T cells that are programmed now to bind to the antigen of their choice. And this has been tremendously successful in rare lymphomas up to now and some myelomas. So CAR-Ts only recognize now cancer cells, only recognize cancer cells. As you can see from here, oops, yeah, pointer is not pointing, but anyway, you can read it. And the 6 CAR-Ts have been approved by the FDA. The price has been reduced allegedly to $500,000 per patient, which is not acceptable if you have a large number of patients, but it can be acceptable if you have so-called orphan drug status, very rare diseases. NICE in the UK also recommended it. NICE was the purpose of the Institute of NICE is to be economic, to be used pharmacoeconomics. So to cut a long story short, CARTs is the first example of a living cell, human living cell, killing cancer. And you can manufacture it and give it back to the patient. So this is a very exciting new chapter in immunotherapy. So to summarize, we have had great success with cancer vaccines, and it's extremely effective and relatively cost-effective. We have great expectations from mRNA and neoantigens vaccines, which is the ultimate of individualized therapy for the poor, for the rich, for everybody. Because the moment a patient comes into hospital, they have their neoantigen screen, and automatically the NHS gives them a vaccine. So you don't need to worry what you call them, whether it's precise medicine or is for everybody, is public. And that's what attracts me with the ESE. ESE is actually a free care system in Cyprus, which is a great achievement. It just needs a bit of fine-tuning. Monoclonal antibodies, of course, we know they are already very important. Checkpoint inhibitors is a huge area that is expanding dramatically. And CAR-Ts, even if one worked, it opens a whole new avenue that we can use human cells to do something extremely useful. So I would remove the question mark, is immunotherapy a reality or not? I think immunotherapy is a great reality at the moment. And we have shown that our immune system is there and waiting to kill the cancer. For the last couple of minutes, I have 0. Can I have another 2 minutes? The last couple of minutes, I'd like to present to you some new projects that we're doing in Cyprus. I mean, I'm at Imperial College in London, but Cyprus is very close. It's around the corner. We want to target tumor cells survival tactics. Why does cancer survive, and what are the weaknesses? We need to find the weakness of the enemy before we do anything about it. What are the weaknesses of the enemy? We are interested in hitting only cells that have mutations, only cells that have driving mutations. There are mutations that we don't know their purpose, but you can find them in cancer. We don't know what they do because we have no knowledge of it, not because they're not important. But there are some mutations we know what they are doing. They are so-called driving mutations. They are important for carcinogenesis, and even not for carcinogenesis, they are important for maintenance. They may be important for spread, metastasis, drug resistance. Those are the nasty ones that we need to hit. To do that, the concept was that we want to develop a protein that is transported, like a Trojan horse. Think of Agamemnon, Achilles, and the Trojan horse, since we have been using a lot of Hellenistic terminologies in the last few days. The Trojan horse is something that goes in and out unnoticed. So we wanted to develop a protein that goes in and out of every cell in the body unnoticed, so they can go into every cell in the body. And we wanted to carry something that is only deadly to mutated cancer cells. So the protein that we're using is Antennapedia. That's why I was very fascinated about Drosophila, because Antennapedia, which is really on the antennae of Drosophila and the fruit fly, is an ancient. We've been talking, in fact, recently, Dimis Hassapis and his colleague shared the Nobel Prize, and because they had sequenced all the human proteins around. And they were shared with somebody else, his name escapes me, who made thousands of other new proteins that we know nothing about. We took a backward step, and we went a few thousand years back, and we looked at a protein known as Antennapedia. You can see it here on the side. It's a 3.5 helices. And on it, at the tip, we can attach anything we like. The things that we like to attach is some things that blocks only mutations. The first example is we're blocking mastermind. Let me try to see if I can point to. Okay. You can see we're hitting this protein in the middle. This is a notch pathway. Simply putting in a cartoon is a notch pathway. And we're giving this drug here, which we call S4. This drug we have made by attaching another human protein, which is called dominant negative mastermind. And our bodies, we have a lot of actions, a lot of pathways, but when these pathways are switched on, they need to be switched off at some point. And one way to switch off a pathway is to use the opposite protein. Think of, for example, even in cancer, we have breast cancer, a lot of it is driven by estrogen. One of the good treatments is an anti-estrogen. So our bodies are really yin-yang. You switch on something, you must switch it off. You cannot have a tachycardia of 100 permanently. As we heard already, you may drop dead. So we're attaching onto this protein, this blue ball, which is dominant negative mastermind, because mastermind, as you can see in there, is the MAML, is the last transcription factor in the notch pathway, which tells that cell, if it's activated, you tell that cell, you're going to senescence now, you go to sleep. If a cancer cell goes to sleep, you cannot kill it. Or you can kill it if you can also kill the normal human stem cells. So if a drug adopts a stemness, that's why I was very interested very much on the notch presentation, or the Drosophila presentation, that if we need to get the cancer stem cells out of stemness. The moment they get out of stemness, they start differentiating and they are sensitive to treatment. So this drug that we developed blocks the mastermind, the notch pathway. And these are some of the early results. You can see in the control, the tumor is a very aggressive triple negative breast cancer, which is it responds to chemotherapy initially, but it may relapse and may be problems with triple negative breast cancer. That's how it grows normally in animals. This is a xenograft. But if you treat it with a product called S4, it shrinks, and at the very end, when you dissect it, it's a small cyst that doesn't contain any malignant cells. And this drug which is S4 and TR4. It has no toxicity that we can see because it just simply blocks very precisely the mastermind transcription factor at the DNA level. So we think this, we need to spend some more work here in Cyprus, but we think this could be a candidate for clinical development. And the second product, of course, is targeting P53. P53 is the commonest mutation in human cancer. And there are more than 100,000 mutations of the P53. But all of these achieve one thing. They disable P53. P53 is the guardian of the human genome. If you have P53, it protects you from cancer. If you lose the ability of P53, then it doesn't, you run into cancer. P53 controls P21, which is the final thing that tells you, please kill a cancer. And many people have been trying to develop anti-P53 products. Nobody has worked, but we have taken the Alexander the Great approach, also the Leon the Governor. So we chop off the thought about P53. We're introducing the wild type P21. This is the work of Christina Kousparou. And she started a great PhD, and look what's happened to her now. She's in charge of everything. So you can see that, I don't know if she's working. So basically, by delivering P21, finishing in a minute, by delivering P21 wild type into P53 mutated cancer cells, they die. And again, this is here, you can see, first of all, in cancer cells, in vitro, it can penetrate every cell and it ends up in the DNA. It's great to have a human protein of your choice to end up in the DNA of a cancer. And,
Professor Benedos, I regret to kindly remind you that we are running out of time, so if you please
just summarize.
Come to the conclusion.
Okay, you don't want me to talk to you about the European Genomics Center. Okay, I will leave that for another talk, since I'm taking your time. I'd like to thank you and say goodbye to you. I left a picture at the top of a black hole. In case Professor Patricios wants to invite me in the future, I can tell you all about black hole, why they are there, what they're doing, and do they represent senescence or immortality. Anyway, thank you very much, everybody.
Thank you. We thank Professor Benedos for his very lively and comprehensive presentation. I think most of us do share your optimistic view for the role of immunotherapy in cancer management. My only concern is the cost and whether this is a limitation for its widespread use and embarrassment by health systems, national health systems. Are there any questions from the audience?
Here, at this point, we would like to thank you, Professor Benedos. Honored to have you. To ask for Dr. Konstantina Stylianou to please share with us her presentation that we skipped previously. Thank you, Dr. Stylianou.
Hello. I want to express my gratitude to Professor Patricios for the warm welcome today. I'm going to try to be quick so we don't miss any time from lunch. So today I will speak to you about a topic that I'm truly passionate about, which is the science of living well with cancer and the role of integrative oncology in cancer care. So during our lecture today, I'm going to try to explain what's the difference between integrative medicine, integrative oncology, in comparison with complementary and alternative medicine. I'm going to explain how does an integrative oncology care looks like in clinical practice, and at the same time, I'm going to review the clinical practice guidelines. So what is integrative medicine? Is this a term that you heard before? Or integrative oncology? Did anyone hear that? Probably not. So it's actually very different with alternative medicine and complementary medicine. Alternative medicine, to begin with, is used in the place of conventional medicine. So that means that the patient will stop any medications they're taking and they will use some form of alternative medicine. Complementary medicine is used together with conventional medicine. However, integrative medicine is also used together with conventional medicine, but in a deliberate manner that is personalized, evidence-based, and safe. So we're not using any modalities that have not been shown in science that are effective. So According to the Society for Integrative Oncology, which I'm also a member, it states that integrative oncology is a patient-centered, evidence-informed field of cancer care that utilizes mind and body practices, natural products, and/or lifestyle modifications from different traditions alongside conventional cancer treatments. Integrative oncology aims to optimize health, quality of life, and clinical outcomes across the cancer continuum and to empower people to become active participants during and beyond cancer treatments. Actually, 40 to 60% of cancer patients are utilizing some form of complementary medicine throughout their cancer journey. And majority of the oncologists are actually not aware of that. This is a study that has been done at MD Anderson, and it states that those patients will likely be women or advanced cancer patients, and they will seek some form of holistic approach of herbs and supplements or even nutrition. So this actually states that patients are seeking for a more personalized and patient-centered approach. So it's crucial to maintain those modalities in a safe environment in conventional cancer treatments. So how does an integrative oncology clinical model looks like? In integrative medicine, we like to view the person as a whole. So we look at their physical symptoms, their social symptoms, as well as the mind-body symptoms. So when a patient comes into our clinic, we assess all their patient's symptoms. We're going to go through their diet. So if the patient is receiving immunotherapy treatment, for example, we're going to discuss gut microbiome, or a patient has low nutrition or can't eat well, we're going to increase their protein intake. We're going to discuss their exercise, as is crucial throughout the treatment, their psychology, and we're going to go through each of their side effects. And then we're going to review the guidelines. So what is the guidelines telling us about? If the patient has pain, we know that through science, acupuncture has been effective in treating the cancer pain. However, if the patient has insomnia, acupuncture does not work. CBT works better, or yoga works better. Or if the patient has hot flashes or dry mouth, a study that's been done on MD Anderson states that radiotherapy-induced dry mouth can be dealt with acupuncture. So at the German Oncology Center, which I'm the head of the department, we managed to set up the first integrative medicine department in the country. We are currently collaborating with some of the best cancer institutions around the world, including MD Anderson, Memorial Sloan Cancer Centers from Israel, and we try to hear our patients and give them a more personalized approach. So, because I don't have so much time, I want to highlight the guidelines that we have been developing. At the organization, the Society for Integrative Oncology, which is an international organization, collaborated with the American Society of Clinical Oncology, ASCO. And we have created 3 guidelines at the moment, which is pain guidelines, fatigue guidelines, and anxiety and depression guidelines. We review all the guidelines on all the research through all the databases, and we found the most effective and safe approaches to be combined during the conventional cancer treatments. For example, if a breast cancer patient is receiving aromatase inhibitor medications, they will develop joint pain. So acupuncture can be effective for that. So we need to find non-pharmacological options to help our patients deal with those side effects they've been dealing on a daily basis. Or if the patient has chemotherapy-induced peripheral neuropathy, which is a very common side effect and they can last for years. And a lot of times we have to decrease the dosages of chemotherapy, even stop certain drugs because of the symptoms. So again, reflexology, acupressure, acupuncture has been shown safe as well as effective. Our next guideline is the anxiety and depression. We know that cancer is a very scary word. So a lot of the patients go through a lot of traumatic, and actually some of them develop PTSD symptoms and the fear of the cancer coming back, or if they're even gonna survive that, they are dealing with insomnia and depression and anxiety. So we have to find modalities to help them overcome those obstacles. And then we know that during active treatments, mindfulness-based approach is very safe to be used, or even yoga, hatha yoga, like mild yoga exercises, hypnosis, relaxation therapy, music therapy, that we can take that to our patients' rooms. Reflexology has been proven safe. And our last guideline that we have developed is fatigue guidelines. Now with the new cancer advances, we have long-term cancer survivors. And we're seeing side effects that we haven't seen in the past. And we can't give our patients, again, drug after drug. So we have, again, I'm stating, non-pharmacological approaches that are safe to be used during the cancer treatments as well as after the cancer treatments. So what is the number one modality that can be used for fatigue? Exercise. Exercise. When I was at MSK, we used to get patients off the chemotherapy chair and we will take them to exercise. Why? Because of this, because of the guidelines. We know that exercise can help resolve the fatigue. Tai Chi. Tai Chi has amazing data that helps with breath work, lung cancer, fatigue, also balance. It's amazing the data we have out there. Why not put them into clinical practice? Why not help our patients deal with those excruciating side effects? Also, maxipassion, that has been mentioned before from our Chinese presenter, is another safe modality to be used. Also, American ginseng, a herb in the guidelines. Wow, I'm so amazed, I'm so happy. So American ginseng is actually been shown to be safe to be used during active treatments to manage the anxiety, manage the fatigue that the patients are dealing with. And this is a summary from all the three guidelines, which divides the major side effects that our patients are going through: pain, fatigue, anxiety, sleep dysfunction, nausea, hot flashes, which they're dealing on a daily basis. So I want to conclude that patients overall seek a more patient-centered and personalized approach. And integrative oncology only uses safe and effective modalities throughout the treatments. I want to thank you. If you have any questions or if you want to seek any support for integrative medicine, please email me, and I welcome any questions you might have.
Thank you, Dr. Stylianou, for your great presentation. Are there any questions regarding? Yes, please.
The microphone, please.
The microphone.
For one of the attendees.
Thank you. Thank you very much for your talk, Dr. Stylianou. Just a small question. How feasible do you think this is within the Society of Cyprus? Because if I'm being completely honest, I've seen a lot of people who would hear the word Tai Chi and think that we're talking about magic. It is a great thing that it exists, but considering that the vast majority of cancer patients, they're of an older age, they do not have this nuanced knowledge that we have. How feasible do you think this would be to integrate in their treatment without them being scared and become even more cautious of everything that's going on?
I agree with you. The Cyprus market can be very difficult to incorporate all these modalities. But at the General Oncology Center, we're doing that. We have Tai Chi classes every Thursday, and we have advanced cancer patients taking part during our classes, which Tai Chi is a very safe modality. But it's very important for the instructor to have oncology training. And I can't stress that enough. And it needs to be in a safe environment. That actions needs to be taken immediately if a patient gets dizzy, for example. But we're already doing that. And I want to make people not be scared and just get the necessary training and start incorporate those modalities, because actually patients are doing them by themselves and without telling them to their healthcare providers.
Okay, any other questions?
This one.
Thank you very much for your excellent talk. I wanted to ask if you have any data on the outcomes of these patients. Do you know if they are truly enhanced by this integrative approach for the quality life years or stuff like that?
There's millions of data out there. And at my department, we have a patient-reported outcomes that we do to each time our patient comes, and we're seeing a lot of improvements in comparison to the patients that are not being referred to our department. But there's tons and tons and tons of research, so we can't say that integrative medicine or these modalities do not work, because the research is out there. It's just this ignorance about the topic.
Makes sense. Thank you.
Okay, I think the session is over, so we proceed to the...
Many thanks to the attendees for your attention and participation.
Thank you very much.
We will now proceed with session 7, State-of-the-Art Therapeutic Surgical Methods, the Robotic Da Vinci and Case Studies. Please join us in welcoming the chairs, Dr. Stelios Kokoulis and Dr. Panos Huntis.
Hello, everyone. I know it's lunchtime, so we want to start the lecture, so we move on. We're going to hear two excellent speakers, Dimitrios Kiparisopoulos and Dr. Savas Chiridis. We'll start with Dr. Dimitris Kiparisopoulos. We're going to talk about the role of robot-assisted surgery for mediastinal disease. Is this a marketing exaggeration or a game changer? I know Dimitris well. He graduated from the University of Thessaly in Larissa. In 2008, he undertook his residency in cardiothoracic surgery at Harefield Hospital. He was trained and established on video-assisted thoracic surgery lobectomies as standard practice for curative lung resections. In 2020, he was honored to have published an article at the Cardiothoracic Net Journal as innovative robotic surgery performed and presented for the first time in medical journal. Dimitris is currently an honorary consultant thoracic surgeon at Harefield Hospital, Royal Brompton, and at Harefield NH Foundation, London. And this is the only robotic and thoracoscopic minimally invasive thoracic surgeon in Cyprus. And he's our associate clinical professor. He's associate clinical professor at the University of Nicosia. Thank you very much, and welcome, Dimitris.
And Panos, I'm associate professor of robotic surgery here in European, not in Nicosia. I used to be in Nicosia. It hasn't been.
That's why.
It's all right.
Yes.
It's all right. Thank you very much, everyone. Before I start my presentation, I need to say that how grateful I am to Professor Patrikios for inviting me here for fourth consecutive year to talk about what I know best, and this is robotic surgery in thoracic surgery. In previous years, we have literally discussed about every potential pathology. I think it's time now to discuss about the kind of cases that, since I did them for the very first time with the use of the robot, I said, I'm not going back. I'm not going to do this any other way. So before I start my presentation, I need to say that I have no disclosure to make. And all this is mainly done here in ESO Athens, where I'm running. I'm the head of the robotic and minimal invasive clinic, just 2 years of age. And since then, we've managed to do double cases than everyone else in the country combined. Just to give you a hint, this is one of the biggest hospitals in Greece, in Athens. I'm sure all women know this is a golden hall. I'm sure every woman in this room knows it. And my office is somewhere there on the 7th floor, where I have clear vision from Iatriko, where Dr. Chiridis is. So I say good morning to him every day, but he never comes back to me. So today, I'm going to demonstrate how things are with a robot, and whether this is worth it or this is a marketing exaggeration, because marketing is everywhere. Is on the TV, is on our job, and unfortunately, it has taken a very big role in our medical practice. So in my institute, apart from doing what I'm going to present you later, I'm so proud that over the past 2 years, we have hosted medical students from this medical school who not only came there to shadow us, but also they let us assist them. So this is one medical student from European University doing a case, simple case, I appreciate that, and I'm helping her. And this is another medical student who put a stent in and opened the closed airway. And this is the last picture from an operation we did at the beginning of August. And this guy here is sitting on the second row. And actually, he can justify, this is the very first ever robotic NAS bar correction for pectus excavatum done for very first time in the world by my team. And I just wanted to say how grateful I was. And these are members not only from medical schools and other medical students from London, but also people from Spain, England, Switzerland, and literally all over the place to be part of this. And these are some pictures from this operation. So marketing, as I've told you, has been to our everyday practice, and I'm sure you'll be waiting for this and that slides that I always show in this Congress. So I'm sure that people will try to convince you that this is Mercedes and this is a BMW, but I can guarantee you that it's not. So let's crack on now and talk about the mediastinal diseases. What is a mediastinum? There are two classifications. This is the first one, but this has literally 4, let's say, chambers, but I'm not so good with 4, so I prefer the one with the 3. So this is a slice of the human body, and this is the spine, this is the sternum, and whatever is in between is divided in 3 parts. The middle part is occupied by one of the most stupid organs that we've got, and we try to kill it. Is beating for a few trillion times during our lifetime, and we're trying our best to destroy it. This is the heart, and that's why there's not so much space there. However, there are some significant pathologies at this area. And the most important one is the anterior mediastinum, and of course, the posterior mediastinum. I remember as a medical student that the pathologies from the anterior mediastinum are the 4 T's. T for thymus, thymomas, thymic carcinomas that are related to myasthenia gravis, T lymphomas, thyroid, goiter or not, or ectopus thyroid, and teratomas. The remaining 5% is a heterogeneous team counting almost 200 different reasons, different pathologies, but the majority of them are related with germ cell tumors. How should we approach I've told you before that, you know, since I started doing my very first case in pathologies like this, I said I'm never going to go back. But am I allowed to do that? And what are exactly the guidelines saying? So if you follow the NCCN guidelines, it actually doesn't say go ahead, man. But believe me, in red pencil, this is the very first time that there is light at the end of the tunnel. The previous one was a definitely no, but now it says, yes, okay, maybe it hasn't been enough data to support doing these procedures with minimal invasive techniques, but due to lack of data. Why is that? This kind of procedures, for instance, the thymectomies, represent 8% of my overall activity. So imagine I'm one of the very few robotic surgeons, not only in Cyprus, but also in Greece. So how can we have enough data to show them that, yes, it's worth it? I will show you later. In comparison to that, it has been officially approved for the past 3 years that the principles of surgical therapy of lung cancer, it has been approved that unless there is any compromisation, and of course, no discussion to the same outcome, a patient should be operated either with VATS or RATS, as well as there is experience. Now, what does experience mean? Who can say that I am an experienced surgeon? I can tell you, you know, I've done 1,000 cases, but Dr. Patrikios will come later and say, you know, he's done 2,000 cases. Of course, you're going to believe him, right? So this is how we try to demonstrate what we're doing and how well we're doing this. This is an operation of mine that has been uploaded, it has been submitted, and of course, announced by the biggest, the most notorious journal in my specialty. This is the CTS-NET, and I'm proud to present the operation that was done for the very first time with the use of the robot. You can see even the logo of the CTS-NET. This is the official web page from CTS-NET. And in this video, I'm showing, you can see with the PET scan, the lesion, where it is. It's a thymic carcinoma, extremely rare. I remember since I was in the UK a consultant, I think I've done 2. And since then, I've done another 2. So it's extremely, extremely rare entity. And as you can see here, this is a robot. These are the ribs. This is a phrenic nerve. This is a heart beating. Not very nice place to work there. This is the lung. And this is a lesion. This is a tumor. So the reason I want to show you this is that at some stage, I noticed that the lesion is very well stuck to the pericardium. So I had a dilemma. What shall I do? Play that I didn't see it, or continue and do the unthinkable, which is what? Just opening the pericardium and excise the mass along with the tumor, with a beating heart just underneath, as you can see, with the slightest wrong movement of my hand or my finger, everything would turn red, and patient would be dead. That operation was done, I think, 4 years ago. The patient is alive and kicking. In this particular procedure, there was no compromise at all on the outcome. It was proved that the pericardium was infiltrated by the tumor, which means if I had left it in, patient would have a different prognosis. That was the first T of the anterior mediastinum. Let's go and have a second T, T lymphoma. You see a nasty lymphoma. This young woman had 4 different biopsies and all turned back negative. Why? Because you see how much necrotic tissue is there. It's not rocket science. We get in, we get samples, it's just a biopsy, and they're very happy because this is the only way to establish diagnosis. Otherwise, the other option would be either to have an open median sternotomy or a Chamberlain, which is still an incision. Would we like that? If it was you, if it was your wife or your children, would you like to have this operation? Definitely not. I'm sure you're going to love this. Let's go to another T. This is a CT of a patient. This is a chest. This is a sternum. And look at this. And look what's happening inside there. You see some calcification there inside it. And this is sitting literally at the top of the heart, at the top of the pericardium. So I'm going to ask our medical students. It's big, it's of the anterior mediastinum, and it has calcification in it. What is the diagnosis? Come on, guys, you know it. Very shy, I'm sure they know.